As was just said, I'm at the La Jolla Institute for Immunology. We are a nonprofit research institute on the University of California, San Diego campus. We're essentially a freestanding immunology department. And what I am generally obsessed with is the biology of B cells and CD4 T cell responses to vaccines. Almost all licensed vaccines work on the basis of protective antibody responses, which are really generated by these cell types. But they almost always completely depend on CD4 T cell help and germinal centers. And that help, we now know over the past 10 years or so, comes from T follicular helper cells. And I put this animation together last year to summarize a number of the things that have been learned by my lab, but also many labs coming together to try and understand TFH biology. In particular, in this case, TFH differentiation and function. And since I'm the first speaker of the group, I'll try and give a real TFH introduction to try and help everything along and hopefully tie things together. So at initial DC priming, naive T cells are TCF1 positive. And depending on the signals they receive, in most situations, those cells pretty rapidly differentiate into either non-TFH cells or early TFH cells, which will then migrate to the T cell B cell border and begin interacting with B cells. And so these TFH cells are important for early B cell events, including early plasma cell events, as well as subsequent germinal center reactions. And germinal centers are a really phenomenal anatomical location, and they completely depend on TFH cells for their function. And a central function of germinal centers is affinity maturation, mutation of those germinal center B cells, and that occurs predominantly by TFH cells being a limiting selective force. It appears in germinal centers. So the TFH are required to provide help to those germinal center B cells in the form of cytokines and CD40 ligand that's required for those GCB cells to undergo proliferation and mutation, and then they have to come back and interact rapidly with TFH over and over and over again. And through that process, high affinity plasma cells and memory B cells are generated in many conditions. And that also, as the GC resolves, it appears that you get formation of memory TFH throughout that process, okay? So as of last year, that's a summary of the knowledge that had been accumulated over the previous decade, including, I guess, that at least in humans, the TFH cells consistently express CXL13, IL-21, IL-4, and some CD40 ligand, okay? And one of the things that's been pretty phenomenal in this field, again, over the past decade leading up to a year ago, was the number of different human conditions for which a role for TFH has been observed. So it wasn't a surprise, given what we knew about the importance of antibody responses, that TFH are important for antibody responses to vaccines, viral infections, bacterial infections, microbiota, and actually Stephanie, the next speaker, really provided critical data about their role in parasites, and she'll speak to I think allergies more today. And there was good evidence that TFH were involved in allergies in the sense of being required for IgE, but unknown how and why, but also involved in a lot of different autoimmune diseases that don't all have identical presentations. And so TFH seemed to be doing driving antibody responses, but also potentially other functions in some situations. And so given those diverse roles of TFH cells in different biomedically relevant diseases, we've wanted to know more about how TFH are regulated, and one of the things that's been central to that is that Bcl-6 is the transcription factor that's required for essentially all TFH cells and germinal centers. And so what I'm going to talk to you about today are the things that we've been doing to try and understand how Bcl-6 functions so that we can better understand when things go wrong or aberrant, why is that occurring, and how does Bcl-6 have roles or not in those in those things. And one scenario is in infectious diseases, and so my lab's been spending a lot of effort trying to understand immune responses to COVID-19, and this is a paper we published with Alex Seti a couple months ago that's been I think very influential in understanding human immune responses to COVID-19. And as we've continued our work in this area, people make substantial CD4 T cell responses to COVID-19 and substantial antibody responses, and we've been trying to understand those and their relationship to disease severity. And we have, of course, looked at TFH responses. This is unpublished data here. And we see that people with acute COVID-19 make TFH responses, antigen-specific TFH responses shown in blue here, and in the convalescent phase, they have TFH as well. And as has been seen in a number of viral infections and other conditions, those TFH cells express additional chemokine receptors in different ratios. And so here, for example, a number of them express CXCR3 or CCR6. And it's been very challenging for the field to understand what those molecules mean and what the heterogeneity amongst TFH cells means. For example, we found that strong SARS-CoV-2 specific CD4 T cell responses are associated with lower COVID-19 disease severity, and including strong TFH responses. So that's saying adaptive immune responses are beneficial in controlling this disease. And there was really a quite strong association between virus-specific TFH cells and lower disease severity, indicating TFH cells probably have an important role to play in that disease. But when you take a look at the heterogeneous markers, there is an association, but it's not so strong, and it's less strong than seen in the non-TFH, i.e. the CCR6 expression. So essentially, we're left with trying to understand, is that heterogeneous marker expression, such as expression of CCR6, important here, or is it just an epiphenomenon of it happening globally of the CD4s, and it's important globally in the CD4s, but not important on the TFH, but they just happen to express it? And that, again, comes back to trying to understand both the heterogeneity of TFH cells and at the transcription factor level, how they are regulated. And so the work I'm going to summarize today is really trying to get at the central question of Bcl-6 is required for TFH differentiation. How does Bcl-6 control that process? What is Bcl-6's role in the cells as a transcription factor? And Bcl-6 has an antagonistic relationship with another transcription factor that's important in immunology called BLIMP1. And generally what we observe are that the TFH cells and germinal center TFH, GCTFH, are Bcl-6 positive and non-TFH BLIMP positive. And these transcription factors directly repress each other. So they generate usually a mutually exclusive gene circuitry to really drive cells to be one cell fate or another, okay? So we went through a process of trying to test several what we considered simple models of how TFH differentiation may be occurring and what the role of Bcl-6 may be. And so a first model was that TFH differentiation might be a default pathway of activated CD4 T cells. A number of people have proposed that over the years. where the model would be basically that Bcl-6 is inhibiting BLIMP1, which is inhibiting TFH differentiation. So if you just, then that's the only role of Bcl-6. And so that model posits that if you get rid of BLIMP1, you should just get TFH differentiation by default. And so we did that by generating double knockouts of Bcl-6 and BLIMP1 in CD4 T cells, and TFH differentiation did not occur. We did this in protein immunizations and viral infections because we know that the role of Bcl-6 is required in essentially all TFH differentiation, so it should occur in multiple models, and so we did that. And functionally, those double knockout CD4 T cells did not become TFH as well. So there were no germinal centers and no TFH cells in the follicle. Okay, and so how does Bcl-6 control TFH gene expression? Well, what's known about this transcription factor Bcl-6 is that it's definitely a repressor. It's definitely known to be a repressor. And so the simplest model would be that Bcl-6 acts primarily as a gene repressor, controlling upregulation of genes via repressor-of-repressor mechanisms. And that's important because we know TFH, most of the genes we're interested in TFH are genes that are selectively upregulated in TFH and actively participate in TFH function or migration or differentiation. So as a repressor, how does Bcl-6 control genes that are upregulated? And so we propose, well, the simplest model is a repressor-of-repressor. So from here, I'm gonna whip through a whole bunch of data pretty quickly and just try to highlight the main conclusions from the data in the interest of time. One challenge has been, is Bcl-6 really primarily a repressor, or is it also an activator? And one of the places people have proposed that Bcl-6 was an activator was actually at the Bcl-6 gene. And that's because people observe a positive association between Bcl-6 expression and Bcl-6 activity. And there is a Bcl-6 binding motif in the Bcl-6 gene, Bcl-6 promoter. And so we tested that, that you can't tell that by correlation. You have to do a direct test. And so we did that by deleting the Bcl-6 binding site in reporter constructs, and Bcl-6 expression went up. And then we went and made mice that deleted the entire mouse's wild type except for an 8 base pair deletion of this Bcl-6 binding site in the Bcl-6 promoter. And the outcome was substantially more Bcl-6 expression. Okay, therefore, Bcl-6 is actually a negative regulator of its own expression, acting as a repressor. And so how do we test this idea of, is Bcl-6 primarily acting as a repressor of repressors? That's a general challenge in transcription vector biology and in immunology, understanding the functions of repressors, because you're basically a step away from the upregulated genes. And so the way we went about doing this was to say, well, we know Bcl-6 and BLIMP1 both repress each other. So if we just get rid of Bcl-6, the problem is a lot of what you then observe is driven by excessive expression of BLIMP1. So the best way to identify genes that are repressed by Bcl-6 and then potentially identify genes downstream of those is to compare gene expression between a Bcl-6 BLIMP1 double knockout and a BLIMP1 single knockout. And so we sorted cells representing TFH and Th1 populations from each of these different genetic conditions. We sorted them and then tried to put all of the data to a test of, did it conform to the simplest gene circuitry model we could posit, which would be that Bcl-6 represses a set of transcription factors that are repressors of the genes that are actually upregulated in TFH and are required for TFH function, such as CXCR5, PD-1, cytokines, etc., as well as Bcl-6 directly repressing transcription factors that are involved in other CD4 T cell fates. Okay, so this is the model to test. And first of all, we could see when we did all this gene expression analysis, we could see that the bulk of TFH gene expression did depend on Bcl-6, because when we deleted Bcl-6, there was a huge shift in the gene expression that was associated with TFH biology, and same thing with BLIMP1. And so then if we bioinformatically analyze all of that data, it actually clustered pretty easily into 4 major gene expression patterns. And so we proposed that 2 of those gene expression patterns would correspond to genes directly repressed by Bcl-6, and then the genes repressed by the repressors downstream of Bcl-6 would be cluster 4. So these are the genes that are upregulated in TFH. All right. Then we also posited that actually to understand that, we probably needed to propose the same thing for BLIMP1. And so the gene expression actually correlates well with 4 clusters that may be genes, cluster 1 directly repressed by Bcl-6, cluster 4 upregulated downstream of Bcl-6 indirectly, and then similar things for BLIMP1. So we tested that by cross-referencing to known ChIP-seq data. And in fact, cluster 1 genes were highly enriched for genes that actually have Bcl-6 binding motifs. Whereas if Bcl-6 is frequently functioning as an activator, the cluster 4 genes that are upregulated in the presence of Bcl-6 should also be bound by Bcl-6, and in fact, they're not. And so these data are actually consistent with Bcl-6 primarily acting as a repressor, and activated genes coming up downstream are because of transcription factors that are repressors that Bcl-6 is regulating. And then we did the same thing with BLIMP1 and saw very much the same pattern, direct regulators versus indirect regulators. So with that bioinformatics in hand, the question then becomes, okay, if this is the main way Bcl-6 functions in controlling cell fate, what are these transcription factors downstream of Bcl-6 in TFH cells that are controlling upregulated genes in TFH? And so we could do that by screening for such transcription factors based on our gene expression profiling, and we did it both in the context of a viral infection and protein immunization, and identified a number of transcription factors of interest. Now, that list was pretty long, and a simple first test was to take one top candidate, ID2, and ask, okay, can one transcription factor basically explain everything? Because we know Bcl-6 represses BLIMP1, and we know BLIMP1 has a role in controlling CXCR5 and some other TFH genes. Maybe Bcl-6 just has to repress these 2 repressors. ID2 is a repressor, and that controls everything downstream in TFH. That turned out not to be the case. We made the triple knockout mice, we made triple knockout cells, and ID2 has a role, but it only controls part of TFH differentiation, and BLIMP1 also only controls part. So to identify transcription factors more broadly, we ran a TAC-seq analysis in combination with our RNA-seq analysis to ask, okay, of all the transcription factors differentially expressed, which of those transcription factors actually have enriched binding for TFH genes that we know get upregulated, such as these genes in cluster 4, okay? There's some transcription factors that are controlling these genes. How do we identify those? Well, those genes may be identifiable by differential TAC-seq patterns in these different knockouts that we did. So we ran that analysis so we did the TAC-seq, ran the analysis for changes in transcription factor binding motifs within TAC-seq regions that changed between Bcl-6 knockout and not, and identified particular transcription factors of interest. There were a number, including RUNX family members, GATA family members, and KLF family members. So then how do we identify which ones are relevant? Well, we can take Bcl-6 ChIP-seq data that we already have from humans and ask, do we have evidence that Bcl-6 directly regulates any of those transcription factors? Because for those transcription factors to matter, they have to be downstream of Bcl-6, but upstream of other genes. And sure enough, RUNX2, RUNX3, and KLF2 fell into that category by the ChIP-seq. Now we have to test those transcription factors and other transcription factors, and making, whatever it was, 35 different knockout mice was not something we were interested in. And so we adapted a CRISPR type approach by directly electroporating in RNPs with Cas9 into TCR transgenic CD4 T cells that we would then transfer in vivo. And this turns out to be a very efficient way to generate knockout antigen-specific T cells in vitro, and we could look at a lot of different functionalities that way. And so we did that. And again, to briefly summarize the data, we could take those double knockout cells, okay, that were normally not differentiating to TFH well, and now we could additionally knock out a transcription factor of interest that we think is a downstream repressor. And the reason for this is, instead of doing it in wild type, is we need to remove Bcl-6 because Bcl-6 would already be repressing this transcription factor. So what we want to ask is, if we get rid of Bcl-6 in BLIMP1, if we also get rid of this repressor, do genes downstream in TFH now increase? And sure enough, for RUNX2, it's a regulator. It's normally a repressor of CXCR5. But it didn't repress all genes in TFH, it repressed a subset. And we did similar experiments with KLF2, and again, like RUNX2, it didn't repress all TFH genes, it repressed a subset downstream of Bcl-6. And in the case of KLF2, it was really interesting because KLF2 was repressing cytokines, IL-4 and IL-21s. And so we take all of this data together, and it does seem to present a consistent pattern that Bcl-6 is central to TFH differentiation and function by regulating repressor-of-repressor gene circuits, and that these are really an interwoven set of gene circuits. And so this is all summarized here, okay? So Bcl-6 is required for TFH differentiation, and one way it does so is to repress alternative fates, such as Th1, Th2, or Th17, and it definitely represses genes like Tbet, GATA3, and RR gamma T. But it also drives TFH differentiation and function by repressing a set of repressors that result in upregulation of TFH genes of interest. And each of those repressors control an overlapping subset of the TFH genes of interest, which can explain why, in the absence of Bcl-6, there's really no single transcription factor that can substitute for Bcl-6 and get TFH. It also explains, I think, how if there's dysregulation in this process in vivo, you can get TFH cells that have aberrant phenotypes because they may end up having disruption of essentially one of these transcription factors and therefore have, for example, a shift in cytokine production, but not other functionalities. And so we hope that this type of gene circuitry knowledge is, for one, just generally valuable for understanding T cell differentiation and differentiation in other systems, that repressor-of-repressor transcription factor circuits can work very well, even if they hurt your brain to try and think about. More importantly, in the case of biomedical diseases, this type of circuitry may help explain some of the diversity of unusual TFH that are seen in diseases, and may help us explain, for example, in the context of COVID-19, what does this heterogeneity mean? And I'll end with this final slide, talking about models for how heterogeneity of TFH cells may occur when, in a fully differentiated TFH, there are very high levels of Bcl-6. And in that situation, Tbet, for example, in a Th1 condition, is repressed and is essentially undetectable. But Bcl-6 expression is not always high. And when TFH go to memory or circulate in the blood, they actually don't have any Bcl-6 different from other cells, okay? So their TFH, the Bcl-6 was required to get them there, but in the memory phase or in the blood, they're no longer high for Bcl-6. And so is it that that's a situation that now that this cell had seen some other stimuli, now that there's less Bcl-6 around, some of those other stimuli, some of those other pathways, such as TBET, can now partially re-express? Or is it one of these other factors that there are bifurcations along the way, which I think is relevant for some of the other discussion today. So basically, I'm just going to say this is my lab, and essentially everything I just showed you was done by Jinyoung Choi, who's a very talented postdoc in the group and who's currently looking for faculty positions as of this month. So I will stop there, and we'll do questions at this point. Is that correct? Is that how you wanted to run it?
Yes, it will be now. So thank you for your presentation. It's very interesting, all the data that you showed. So we have a question from Thomas Tiuchi from University of Rochester. He says, thank you very much. In the model where Bcl-6 represses its own expression, wouldn't you expect effector TFH to preferentially become long-lived non-TFH cells?
Say that one more time.
Yeah, so in the model where Bcl-6 represses itself, wouldn't you expect effector TFH to preferentially become long-lived non-TFH cells?
Long-lived non-TFH cells?
Exactly.
No, I think the simplest model would be, since Bcl-6 is normally repressing itself, if we get rid of that repression, Bcl-6 will be expressed more and expressed longer, and so there would be more TFH and more memory TFH. Connecting to what I just said a minute ago about, Bcl-6 itself expression is not stable, right? It's required for TFH, it's key for TFH, but it comes down again in memory and in a couple other stages. And so there are, I'm not showing it in this diagram, right? But there are clearly upstream signals that are driving active Bcl-6 expression. And so it's that combination of the signals driving Bcl-6 expression during any given time window, combined with Bcl-6 autoregulation that gets you, I think, the amount of TFH differentiation you get and the stability or instability of the TFH differentiation, right? I think this helps explain why Bcl-6 is not retained going from GC-TFH to memory TFH.
Okay, another question from, thank you. Another question from Eugenio Contreras, who's the host of the Open Box Science from Mexico. And in this program, is this program only required for differentiation, or is required for maintaining the TFH phenotype? What would happen if you performed experiments in a Cre-ER2 system?
Yeah, it's a really good question. I mean, the available data are definitely that Bcl-6 is continuously required for differentiation and function of TFH cells, and that's work, published work from other groups. We haven't published our data on that. In terms of whether transcription factors downstream, right, Bcl-6R transcription factors are required just in the differentiation stage or in the function stage, I think it's going to vary depending on the transcription factor. We don't have a great way to do that experiment at this point because we can't do essentially partial conditional Cres or introduce the Cas9 well introduce the RNP well at a later time point, sorry. But I would expect, for example, let's say KLF2. So KLF2 is involved in migration of cells, okay? And so KLF2 may be important to repress by Bcl-6 early because KLF2 drives CCR7 and CD62L expression, right? So if you can't repress that, the cells are probably going to have difficulty differentiating to TFH if they sustain these. But then, in terms of Bcl-6 and in terms of TFH function, KLF2 is it looks like needed to get IL-21 and IL-4 expression by the TFH, which is going to be a later activity. So I think both, for example, for KLF2, it would be both early and late. For the other ones, we're not sure. A very good question.
Okay, another question from Jose Crispin. from Mexico City as well. And he says, great work. Do other TF transcription factors participate in modulating the inhibition of the downstream TF by Bcl-6?
Yeah, that's a great question, and I think an important one to bring up. We are not arguing that Bcl-6 goes it alone. We're not arguing that Bcl-6 is the only transcription factor that's doing anything in TFH cells. But what we do know is that it's required, and it's and that it's essential. There are a number of other transcription factors that are required, such as TCF1 and LEF1 upstream, and we don't know how all of those things, we don't know how all of those things piece together.
Okay, thanks. And there's another question from Marco Tapia Maltos from Mexico City as well. And thank you for your presentation. In the paper, you mentioned that there was another transcription factor that binds to the Bcl-6 promoter in a site that overlaps with the Bcl-6 binding site. Instead of repressing its own transcription, Bcl-6 binding could interfere with the binding of the other repressive transcription factor and stabilize its own expression?
Yeah, that's a really good question. That definitely is a formal possibility. There's really no good way for us to exclude it. Anytime you're going to manipulate a promoter, you have the possibility for sure of affecting additional things. The overlapping site was a STAT site, which would be expected to positively regulate the Bcl-6 expression based on STAT. So what we can say is we made the smallest deletion we possibly could, right? Whereas most people are deleting like 500 base pairs or 4 kb and saying that there's an effect on transcription. We literally deleted just a couple of base pairs. And we think the data is most consistent with this, because if it was disrupting the other transcription factor, we expect the primary outcome would have been the opposite outcome. That's basically it.
The last question, could the repressor function of Bcl-6 could be altered in non-responders to vaccines?
It could, but we haven't seen. So one of the ways, there's a huge amount of human genetic diversity information available, right, in the form of SNPs and essentially EQTLs, and there's no strong evidence to date of Bcl-6 itself playing that role. And even if it is, it would be very hard to distinguish whether that role was occurring in the CD4 T cells or the B cells, right? Because Bcl-6 is both required for the TFH and the germinal center B cells. So we suspect some of that variation, some of the variations seen in humans probably does come from variations in TFH differentiation and CD4 T cell biology, but may come not from Bcl-6 itself, but how it's regulating downstream transcription factors and genes, right, or variation in those. That's there are a number of, for example, SNPs of interest in BLIMP1 that may have functionality in different disease states, but also BLIMP1 is expressed in a number of different cell types. And so none of that has directly tied back to TFH biology at this point.
All right. Well, thanks a lot for the presentation. And I think we're going to continue. Alisa, do you want to take over?
Yeah, thank you, Florencia, and thank you, Shane, for your presentation. I have the pleasure to introduce the next speaker, who is Stephanie Eisenbarth. Stephanie is an associate professor at Yale Medical School and has had a very fruitful career in unraveling how dendritic cells direct specific types of T cell responses, including T follicular helper cell dependent antibody responses in the context of allergy pathogenesis. Stephanie started her scientific career with a background in inorganic chemistry at Bryn Mawr College, after which she moved to study medicine at Yale University School of Medicine. And during this time, she did her PhD in the lab of Dr. Kim Bottomley, where she investigated the role of toll-like receptor-dependent Th2 responses in asthma pathogenesis. After her residency, she went on to do her postdoctoral training with Professor Richard Flavel, where her work focused on the role of node-like receptors and dendritic cells in shaping the adaptive immune responses. She now has her own lab, and they're currently focusing on how the interactions between dendritic cells, T cells, and B cells induce adaptive immunity, with a particular interest in how these interactions contribute to the pathogenesis of food allergy. And now she's here to talk to us about her latest work on the divergent TFH cell requirements necessary for IgA and IgE production to peanut. So welcome, Stephanie, over to you.
Thank you very much. And I really do want to thank the organizers for putting this incredible series together. I think this is a really neat idea. And I think it's awesome to bring together a group of TFH biologists to do a session. Shane did a beautiful job of introducing TFH cells and going into molecular details on them that are crucial to understand how they function. This is going to be a slightly different talk in which